First, I want to thank the FFB and OIS for the opportunity to talk to you and tell you about Wave Life Science and our Stereopure oligonucleotides. We're a publicly traded company, and as such, this talk may contain forward-looking statements. So we're a clinical stage genetic medicines company. We're in the clinic in the neuromuscular and CNS space already. In the neuromuscular space, we're utilizing exon-skipping oligonucleotides that are stereopure to skip exon 51. In the CNS space, we're using stereopure oligonucleotides that are allele selective for Huntington's disease. We selectively knock down the mutant allele while maintaining and leaving the wild-type allele intact. I'll talk about ophthalmology and how we use those, both of those technologies. Prism is our proprietary platform. Most ASOs companies can optimize their drugs based on sequence and chemistry. We had an extra layer, stereochemistry. Stereochemistry refers to controlling the phosphorothioate during synthesis. So oligonucleotides have used phosphorothioate linkages for years to enhance in vivo uptake. But phosphorothioate linkages have a chiral center due to the fact that there's a sulfur, which means in a stereorandom molecule, an ASO that's on the market or that's in development to date, is actually, if it's a 21-mer, it has 2 to the 20 permutations because every single linkage between every nucleotide can mean one of 2 conformations. At Wave, we control every single linkage. Therefore, our oligos are single diastereoisomers. What this means is that in vivo, in the eye, stereopure oligos shift potency. So in the upper left, you see a dose response looking at the back of the eye with a stereorandom molecule in black targeting MALAT1, a long non-coding RNA. We have dose response activity, but when we make it stereopure, we shift the potency. 5 micrograms of the stereopure is as potent or better than 50 micrograms of the randomer. In the center, you see an improved PD-PK profile. So in the blue circle, you see that lower drug in the tissue results in greater knockdown of the target. And we have improved duration there in the upper right. 5 microgram of the stereopure molecule holds potency at 3 months, and you see 50 microgram, the pure gives better knockdown. In the bottom panel, you'll notice that when we go to large animal, this holds. So the top is the mouse eye. We just take the back of the eye. That includes retina, sclera, choroid. In the bottom, you see non-human primate. Here we can pull the retina. When we pull the retina, we have complete target knockdown and visualize in red the oligo throughout the eye at 4 months. Now, we utilize those technologies for allele selectivity for a couple of programs. One, our early programs that we're working on are adRP and focused on the P23H mutation. On the left, you see a stereorandom molecule pulled from published patents, which is claimed to be allele selective, yet you'll see it knocks down both wild type and mutant. But on the right, when we make a stereopure molecule, the wild type allele is completely intact. We knock it down 0, but we have complete knockdown of the mutant allele. For our Usher program that we're working on, we have this is an exon-skipping approach. On the left, you see on the left, you see gymnotic delivery, which means no delivery vehicle to 2 different cell lines. There's a molecule that we pulled from the literature and patents as well that's stereorandom. Here we get 5 to 8-fold increased skipping efficiency in vitro. On the right is human target engagement. We took human cadaver eyes, isolated the retina, cultured them, treated with stereopure oligos, and we have 25% skipping. In summary, oligonucleotides that are stereopure give us broad distribution across the retina, all cell types of interest, and allows us to target both recessive and autosomal dominant diseases. Thank you.